This is a production of Cornell University.
I'm Larry Smart. I'm a professor in the Horticulture section of the School of Integrated Plant Science, based here at Cornell AgriTech in Geneva. So at Cornell, we started hemp research in late 2016. We have been doing variety evaluations, assessment of disease threats and insect pest threats, and doing some work on crop management, especially disease management and weed management. So with legalization of hemp in the 2014 Farm Bill, Cornell eagerly took up the challenge of starting to work on this crop that has not been studied for decades and has a high regulatory burden for us as researchers. We have been very fortunate at Cornell to have active engagement across multiple disciplines to build a engaged and diverse team of researchers and extension specialists, including myself as a plant breeder, Joss Rose doing biochemistry of cannabinoids, which is critical to maintain compliance and to breed compliant cultivars, Chris Smart and Gary Bergstrom in plant pathology, evaluating disease threats, and Neil Matson working on controlled environment production. Most recently, Lynn Sosnowski here at Cornell AgriTech has joined our team working on herbicide efficacy and weed management. Alan Taylor is our seed scientist, studying seed quality and working on seed coatings. We have been very fortunate to have a strong collaboration with New York State Department of Agriculture and Markets, who has given us quite a bit of latitude for what I would call self-auditing and self-regulation. And because of that, I think we have been able to make some major breakthroughs in our understanding of the genetic basis of THC and CBD production, which has contributed to rapid breeding for THC compliance and truly an understanding of the genetic basis for THC production and THC compliance in hemp crops. We have also gotten strong support from New York State, with strong supporters in the legislature and the governor's office, to initiate the first land-grant academic hemp breeding program. So we have been breeding hemp varieties, building a hemp germplasm collection, and developing markers for marker-assisted selection since early 2017. Our phenotyping is largely based on the evaluation of current commercial cultivars in 3 market classes. We are very interested in developing new cultivars for grain production, and the grain will meet a niche for animal feed, particularly organic animal feed. We see strong potential in the poultry market for hemp grain improving egg quality. It is a gluten-free feed, so we see strong potential for beef and actually pet food production. But it also is a source of protein and potential ingredient source for plant-based meats. And also it produces a very healthy oil, which is also used in health and cosmetic products. So there is a strong potential to increase yield of grain production. We're also very interested in the fiber market, and this field behind me is evaluation in strip trials of some fiber varieties from China, which flower extremely late, which makes them very good biomass and fiber producers. We see strong potential in fiber production, not only for the long, strong bast fibers, which can go into technical applications, reinforced panels, building materials, insulation materials, for example, but also for the inner hurd, the inner core of the stem, which has potential, again, for construction materials in the form of hempcrete, but also serves as a very good absorbent product for animal bedding. And in fact, that is one of the immediate products that has gone onto the market, is for small animal bedding for your gerbil cage, for example. A lot of potential for further development of products, for example, absorbent products for diapers, for environmental cleanup, and other applications. So once we have sufficient agricultural production of hemp, I think we're going to see great market expansion and diversification of the products that we can make, both from the grain and from the stem. A lot of the hype around hemp has been based on the production of cannabinoids, primarily cannabidiol, or CBD. So we saw a huge interest in 2019 from farmers after cannabis sativa was taken off of Schedule 1, hemp was taken off of Schedule 1 by the 2018 Farm Bill. That released a lot of the regulatory issues and encouraged farmers to take up the production of hemp, but that resulted in extreme overproduction. Estimates that we produced as much as 4 or 5-fold more biomass than the market could bear. And in fact, there are still growers who have material in storage that has not been fully processed from the 2019 growing season. So prices crashed, and we see this sort of classic characteristic roller coaster ride of over-enthusiasm, overproduction, and then a crash of prices, a disillusionment, and now we are in the process of diversification of the market and re-engagement of growers as we continue to improve production practices and develop new varieties to meet those needs of the market. So the Farm Bill authorized us to do research on hemp. Hemp and drug-type cannabis are a single same species, Cannabis sativa. In fact, through some of our work, we have identified that there's a rather simple genetic basis for either high THC production, relatively equal mixed production of THC and CBD, or high CBD production. The interesting phenomenon that we've characterized in CBD hemp is that the enzyme CBDA synthase also produces a certain amount of THC. So every CBD hemp plant also makes a little bit of THC. We at Cornell only work on hemp, which is cannabis sativa that produces less than 0.3% THC. Those regulations have been a patchwork from state to state, and again, New York State has been a rather compliant state to grow hemp in, because their regulations have been based on quantification of delta-9 THC rather than total THC. These are some of the complications and regulatory burdens that we face in working with hemp. We've also been very fortunate at Cornell AgriTech to receive strong support from USDA ARS, and in fact, through initiatives from Senator Schumer's office, ARS decided to locate the new hemp germplasm repository at Cornell AgriTech in Geneva. So that will be a tremendous advantage for us in our breeding program to be able to work side by side with the curator of the Germplasm Collection and geneticists characterizing that collection. We will work with them to assemble material, make sure that it is compliant for THC so that it can be entered into the repository, scaled up, and then distributed to hemp breeders. So for every other crop that academic and industrial breeders work on, there is an extensive and well-characterized germplasm collection that used to exist for hemp, was assembled back in the 1920s and 30s, but then was destroyed when hemp was put on the controlled substance list. So Right now we face the process of reassembling a diverse germplasm collection and characterizing it using modern phenotyping and genotyping tools so that there is useful and diverse germplasm ongoing into the future for industrial and academic hemp breeders. So this is our 4th year of evaluating cannabinoid hemp varieties. In this trial behind me, we have 40 different CBD and now cannabigerol CBG varieties that we evaluate in small plots, 5 plant plots replicated 4 times in the field. We're using typical production practices for the Northeast, which is to plant into raised beds with plastic mulch for weed management. We put down landscape fabric in the alleys so that we don't have to mow, and also to provide a nice uniform background for drone imaging. So we will be doing active weekly drone imaging of this trial to be able to quantify nutrient status, any disease and pest incidents, initiation of flowering, and to use LIDAR to estimate biomass production and yield. By evaluating these cultivars for the last 4 years, actually we have identified trait segregation because the state of hemp breeding is still in its infancy for CBD production in the US. So actually many of the quote-unquote cultivars that we have evaluated have been segregating, some of them segregating dramatically for single major gene traits, such as flowering time. So we have been able to map and identify candidate genes that determine day neutrality. Hemp is a short day plant, but does have some variants that are day neutral that will flower after a certain maturity date. So that is a simple single gene recessive trait that is segregating 3-to-1 in F2 populations. We are also very interested in mapping genes for powdery mildew resistance. Powdery mildew is one of the diseases that has been most abundant, most prevalent in the Northeast and in New York. It's also an important pathogen in controlled environment production. So if we can identify genes and molecular markers to introgress powdery mildew resistance, it would have a big impact on the industry. By evaluating cultivars year after year with Chris Smart and her lab doing the powdery mildew evaluations and characterizing powdery mildew populations, we have been able to identify some varieties that show consistent, strong resistance and have developed F2 mapping populations, which we have in the field this year, hopefully to identify genes for either susceptibility or resistance to powdery mildew. We will also be sequencing the Golovinomyces genome once we can get sufficient high-quality DNA. So in addition to mapping major genes for powdery mildew resistance and for the day neutral trait, we are very interested in quantifying the quantitative differences in critical day length. So there is quite a bit of variation in flowering time from late July, early August through early October, which represents adaptation to different latitudes, and being able to develop cultivars with the proper critical day length and flowering time sensitivity for different latitudes is a big issue right now. For example, there is a lot of interest in the Southeast and in Texas in fiber production, but most of the fiber varieties have been developed at northerly latitudes, so they flower very early when we bring them to southern latitudes. Also, there are tropical countries that have been growing fiber hemp forever, but do not have access to high CBD hemp, which have been developed by crossing hemp with high THC cannabis and then selecting for high CBD production in those effectively hybrids. So there are folks at equatorial latitudes that are very interested in having high CBD hemp, which can grow on a 12-hour cycle. That's a problem that we have not solved yet. Another trait that we're very interested in characterizing is the relationship of cannabinoid production to insect damage. And it has long been theorized that hemp produces cannabinoids, THC and CBD, potentially for insect defense. We have a little bit of insight into that because there are cannabinoid-free grain and fiber cultivars, and we have seen that those particular cultivars seem to be heavily damaged by herbivory from Japanese beetle. So this observation has led us to test the hypothesis that the production of cannabinoids plays a role in pest interactions, perhaps as a defensive agent or perhaps in the deterrence of pests from herbivory. We have developed a mapping population that has variation in cannabinoid production. Some plants are cannabinoid competent, some are cannabinoid deficient, and we have planted those on multiple sites and will survey for relative damage by insect pests in the field.
This has been a production of Cornell University, on the web at cornell.edu.